Good morning, AP Biology students, or good afternoon, depending on what time of day you're watching this. Today we're going to be studying embryology, the development of animal embryos, and this relates to our recent study of gene expression, the regulation of gene expression, the ability of cells to control which genes are turned on, being transcribed and translated, versus which other genes are being switched off. And because part of embryological development, a very big part of it really, is a process called cell specialization or differentiation, where stem cells turn on certain genes and turn off other genes to kind of set their destiny, if you will, to sort of decide what those stem cells are going to develop into. Are they going to give rise to nerve cells or skin cells or muscle cells or liver cells? And so this very much relates to what we've been studying. But you can see we're kind of hopping around a bit in the textbook. So to prepare for this lesson, before you go any further, stop and make sure you read page 322, just that one page, and then skip ahead, way ahead, to pages 764 to 767 in the Campbell Biology in Focus textbook. Okay, so let's get started. So we're going to begin in this diagram right here with a zygote, which, as you know, in animals would be a diploid fertilized egg. It is the union of a sperm and an egg at fertilization, giving rise to this single diploid cell. And the very first thing that happens in the development of any animal's body, including your own, is that zygote goes through cell cycle. It grows. It makes a copy of all of its chromosomes in the S stage, and then it prepares to divide, and then it does mitosis and cytokinesis, and it divides to make two genetically identical cells. And they also divide again to make four. And you can see this early stage of cell division is referred to as cleavage. It's just rapid, repetitive cell division, where the zygote divides to make two cells, which divide to make four cells, which divide to make 8 cells. We're looking at the 8-cell stage. If we could see one frame past this, we would see the 16-cell stage, which is technically the defining point of what we call the morula. A morula is just a fancy term for a solid ball of genetically identical cells. And at this stage, every cell in this ball would be considered an embryonic stem cell. It's what we call a totipotent or pluripotent stem cell, meaning everything is a possibility. This cell can give rise to all the different tissue types in the body. In fact, if we were to just take one cell out of this ball of cells and pluck it out and separate it from the rest, that single cell would have the ability to develop into an entire animal, an entire organism. So the morula is the solid ball of cells right around the 16-cell stage, and it continues to be a solid ball of cells until it gets up to about the 256 cell stage. So again, we're just doubling, right? So 16 becomes 32 cells, which becomes 64 cells, which becomes 128, which then doubles to become 256. And at this point, in the middle of this ball of cells, it's no longer a solid mass. You can see that there's a hollowing out that begins to take place. You have this fluid-filled cavity in the middle of this ball of cells. Now the ball of cells, the hollow ball of cells, is referred to as the blastula. and the space in the middle is called the blastocele. But at this point, all of the cells are still totipotent or pluripotent embryonic stem cells. So the same thing I just said still applies. If I was to take just one cell and pluck it out, separate it from the embryo, and let it develop, it would develop into an embryo. It would develop into a child, an offspring, but because this is not just for humans, this is for all animals. And in fact, that's essentially what happens naturally in the case of identical twins. Identical twins are the result of a very early sort of random separation of embryonic cells. So maybe at the 2-cell stage, or I guess it could be later, like in the 8-cell stage, you have one or more cells which separate from the embryo and develop into a separate organism, which of course is genetically identical to the original. So it's sort of a natural cloning that happens sometimes. Now, as this blastula, again, a blastula is a hollow ball of embryonic cells, as the cells continue to divide, notice a strange thing starts to happen. At one end of the embryo, there's a folding in, what's called an invagination or a pouching of the cells, where they literally fold inward to create a little pouch. And that pouch will eventually be known as the archenteron. You can see it when it's a little further along right here. The archenteron is the very beginning of what will become the digestive system. So in your body, this hollow space right here that forms during this early embryonic development, this hollow space called the archenteron will become your esophagus, stomach, small intestine, and large intestine. It will become your digestive tract. Now, a couple other points I want to make before I leave this image. This picture is pretty well drawn to scale. Obviously, they've magnified the cells to make them look really big so that we can see them. But when I say it's drawn to scale, what I mean is notice that the 8-cell stage embryo and even the 256-cell embryo is almost exactly the same size as the original single cell. So how is that? How can you have an embryo that has 256 cells and it's no larger than when it was just one cell? How's that possible? It's because cleavage is happening so fast at this point. Cell division is happening so frequently that there's not enough time for a G1 stage of cell cycle. So the cells are not growing. They're just dividing. And notice the overall size of the cells is getting smaller. And in humans, at least, in human embryos, that process takes place for about the first 15 days, so a little bit more than 2 weeks, the first 2 weeks of your life. You were in this rapid cell division stage. Now, right around the 15-day point for human embryos, as the archenteron is folding inward even more to make this pouch, another really important step begins to take place. And that is, now we can no longer say that every single cell in this embryo is totipotent or pluripotent, because the cells have begun to specialize. So if I was to pluck out one of these cells right here that's color-coded in yellow, that cell can only and will only give rise to digestive system, organs and tissues, maybe a few other things, but it'll never give rise to skin. It'll never give rise to nerves. On the other hand, if I was to take one of these blue cells, which is known as the ectoderm, those cells can never give rise to digestive system. They are more specialized now, and they can only give rise to things like skin, and body cavity coverings and nerve tissue. So what am I saying? I'm saying that during this process called gastrulation, for the first time in your life, your cells began to specialize. And in most animals, you're eventually going to develop three distinct layers of cells at this point. The ectoderm, which is going to be this outermost layer of the embryo, the endoderm, shown in yellow here, which is the innermost layer of cells, and then there's a third layer that's not shown in this picture, but we'll get to it in a minute, called the mesoderm. So I'll say more about that in just a bit. At this point, I'm going to pause and give you today's code word for extra credit. Let's give a big shout out to this year's class of 2020, the graduating class this year. Today's code word is class of 2020. And my hat is off to them. This is a tough time to be a senior, but we wish them the best. Today's code word, class of 2020. Okay, so let's define some terms very quickly. You know what a zygote is, a single diploid cell that's produced by fertilization, sperm and egg fusing together. We talked about cleavage, which is the rapid multiplication of embryonic cells when they're just dividing by mitosis. Okay, I mentioned embryonic stem cells. I've mentioned the terms totipotent and pluripotent, which are kind of interchangeable terms. Embryonic stem cells are undifferentiated. That means they have not specialized for any particular function yet. Genetically identical cells that are produced by that early stage of cell division, that cleavage, that rapid mitosis. These cells are totipotent, or sometimes you'll hear the term pluripotent. That means they have the ability to give rise to any germ layer, ectoderm, endoderm, or mesoderm. I'll talk about those more in a minute. And therefore, they can give rise to any tissue type in the body. So an embryonic stem cell can give rise to nerve tissue, muscle tissue, blood tissue, connective tissue. In fact, a single embryonic stem cell still has the ability to develop into an entire organism. You saw the morula, which was the solid mass, or the solid solid mass of cells, kind of the early stage of embryonic development. So this was actually the 8-cell stage. I think technically the 16-cell stage is where you consider it to be a true morula, but it's a solid ball of cells. You saw the blastula. Let me go back. When it becomes sort of hollow in the middle at about the 256-cell stage, we call that the blastula. So you go from a zygote to a morula to a blastula. And so this is a hollow ball of cells produced by the mitotic division of morula cells. And there's a fluid-filled space in the middle of the blastula, which is called the blastocele. The process of gastrulation is when the blastula turns into a gastrula. So what does that mean? It's where you go from just being a hollow ball of genetically identical, well, not just genetically identical, but identical in every way, cells, to being a more sophisticated ball of cells where you actually do have some cell specialization. So a gastrula is somewhat of a hollow ball of cells, but it has multiple different layers of what we call germ layers, ectoderm, endoderm, eventually mesoderm. And so the difference is in a gastrula, the cells have started to specialize, whereas in the that's the gastrula. In a blastula, all of the cells are still totipotent embryonic stem cells. Let's see, cell differentiation is the process in which stem cells give rise to specific cell types that have a specific purpose. And that's sometimes called cell specialization. All right, and I wanted to go back to this concept. I mentioned this weird word, archenteron. When you developed from a blastula to a gastrula right around about the 15th day after conception, and that pouch began to fold inward. That was the beginning of the development of your digestive tract, what would become your esophagus, stomach, intestines. And that pouch is called the archenteron. The cells that form the lining around that pouch are your endoderm cells. And I want to point out something very important. The archenteron, this digestive pouch, initially only had one opening to the outside world, which is called the blastopore. Now, if you think about it, in your body now, your digestive tract has two openings, hopefully, a mouth at one end and an anus at the other. Most animals have a digestive tract with two openings. And you say, well, what animals don't? Well, I might have a picture here. Let's see. Yeah. So there's two groups of animals, phylum Cnidaria, which is jellyfish, sea anemones, coral, the little animals that form coral reefs, and also phylum Platyhelminthes, which is the flatworms, like tapeworms. Those two groups of animals, jellyfish and flatworms, never develop past the archenteron, basically. In other words, they live their entire lives, even as adults, having only one opening for their whole digestive system. Notice this opening is a mouth and an anus. So that sounds bizarre, but that means that a jellyfish, when it takes in food, the food goes into this, which is called the gastrovascular cavity, which is basically the archenteron. Now that it's fully developed, it's almost like a stomach. The food is digested, the nutrients diffuse out into the surrounding body tissues, and then the digestive waste is released out the same opening that the food went into. So it is a mouth and an anus. Thank goodness we are not in that animal group. But most animals, everything that's more sophisticated than a flatworm, which is pretty much any animal you can think of, has two openings in the digestive tract. But you didn't start out that way. You started out with only one opening called the blastopore. And for humans, that opening right there became the anus, or becomes the anus, and then the mouth develops at the other end later on. So more about that little exciting story in a bit. I've already kind of talked about this. The gastrula is this stage, right, for us humans. It's right around the 15-day point. It's an early stage embryo having three distinct germ layers. And notice it says sometimes two. In jellyfish, they only have endoderm and ectoderm. They never develop the third germ layer, but we're going to talk about the fact that most animals, anything more sophisticated than a jellyfish, actually develops three distinct layers of cells. These are called germ layers. Any animal that has three germ layers is called a triploblastic animal. So the terminology gets kind of crazy here, but basically you go through this process called gastrulation when your cells do start to specialize, and in most animals that results in three distinct layers of cells, which then develop into all of your different tissues. So I'm going to skip over some of this because we've already talked about everything on this slide. So I've been sort of hinting at this, but at the gastrula stage, most animals, everything more complex than a jellyfish, does in fact have three distinct germ layers. And you can see these are color-coded in these drawings at the bottom. So if we look at this picture here, everything you see in blue is ectoderm. That's generally the outer layers of the embryo. Yellow is the endoderm, which will become your digestive tract. And notice initially there's just one opening. Remember, that's called the blastopore. and then a second opening will eventually form. So this basically this pouch, the archenteron, gets deeper and deeper and deeper until it eventually connects over at this end, and you end up with a tube. There was a point in your life, right around the 15, 16, maybe 17-day point in your life, 17 days after conception, where you were nothing but a ball of cells with a tube running through the middle. And that tube became your digestive tract. Those cells are called endoderm cells, and then the red is the third layer, which is the mesoderm. This is the point where cells do start to specialize. Certain genes get switched on, other genes which are not going to be needed get permanently switched off. And it's different genes in different cells getting switched on and off, which is why some cells become ectoderm cells and other cells become endoderm cells. Every cell in this embryo has the exact same genes, but there are different genes switched on and different genes switched off. So ectoderm cells specialize so that they can only develop into skin and other types of coverings in the body, like the linings of your body cavities and your nervous system. Kind of interesting that the same cells that give rise to skin also give rise to spinal cord, brain, and nerves. You wouldn't necessarily have assumed that, but that's the case. Endoderm develops into your digestive tract and also the lungs. And then the mesoderm is what forms basically everything else. All of your internal organs, your muscles, your bones, your reproductive system, and most other internal organs develop from this third layer, which is the last layer to form, which is called the mesoderm. So this is just a little more detail of what I was just talking about. Here's ectoderm, mesoderm, endoderm, and you can see it breaks it down to all the different body parts that form from the ectoderm, all the different body parts that form from the mesoderm, which is a lot because it's most of your internal organs. Notice endoderm, epithelium, that would be skin, that forms the lining of your respiratory tract, and then of course all this is digestive, mostly digestive system. So generally speaking, you can think of ectoderm as skin and nerve, endoderm as digestive system and part of your respiratory, and then mesoderm is pretty much everything else. So some pretty cool pictures here. This would be a zygote. Notice this would be the 4-cell stage, and it's kind of hard to count these, but I'm assuming this is about the 16-cell stage. So this would be the morula, the solid ball of cells. Here's a blastula. Notice the hollow blastocele. So this is about the 256-cell stage of a human. Notice it's almost exactly the same size as the zygote. And then here's gastrulation. We start to see the archenteron right there folding in. So this would be the blastopore right here. And then this is that same gastrula a little bit further developed, where we can start to see the entire digestive tube forming. And the next step after this stage is called organogenesis, where the endoderm and the mesoderm and the ectoderm actually start to develop into tissues and organs. So a couple of weeks after this, the embryo starts to look a little more like this. It's a pretty big transformation. When those cells start to specialize, the body really starts to take on a shape that you can start to recognize as an animal, or in this case, a human. Organogenesis means creation of organs. So this is, as I said, just the actual development of your germ layer cells into the organs of the body. Some of this is just review. We've already said that muscles and most of the internal organs develop from the mesoderm. I thought this would be a good time to clear up some confusion that you may have. A lot of people are confused about the different types of stem cells. I've already mentioned stem cells. I've talked about embryonic stem cells, and I've used these two synonyms, totipotent and pluripotent. But I want to make sure you understand the different types of stem cells. Okay, so embryonic stem cells is the first major type of stem cells. I'm showing two different types here. There's actually three types. I'll mention the third one briefly, but embryonic stem cells are those early stage cells in the life of an embryo before gastrulation begins. In other words, before any specialization of cells has taken place. So it's either a single cell, or it's that solid ball of cells, or it's the hollow ball of cells like the blastula, where you just have 256 identical cells. None of them are skin cells, none of them are muscle cells or nerve cells. They're just cells. And at that point, they are totipotent, or the synonym pluripotent, which means each cell can give rise to any tissue type in the body. And that's because no genes have been deactivated. But once the germ layers are formed, and you get to the gastrula stage, and you have a distinct endoderm, ectoderm, mesoderm, from that point on, the only stem cells in that embryo, in that organism, are called adult stem cells. This is a really bad term because it sounds like we're only talking about cells in an adult, like you or me. And that's not really what we're talking about, because even at the gastrula stage, when you're only 2 weeks after conception, the only stem cells really in the embryo at that point are these so-called adult stem cells. So that's a really bad term. I've noticed some textbooks have started referring to them as somatic stem cells to differentiate them from embryonic. But certainly at this point in your life, the only stem cells you have remaining in your body are adult stem cells. And these are scattered throughout the body, and you're going to keep some of these adult stem cells in the tissues of your body for the rest of your life, and they serve a really important function. These are the cells that can generate new tissues when we need to develop new tissues, when we need to heal from a tissue injury, when we need to form new blood cells, for example. The cells that make that possible are adult stem cells. But here's the big difference. Adult stem cells, or remember the new term, somatic stem cells, are multipotent. They are not totipotent or pluripotent. They cannot give rise to any and all tissue types, but they can give rise to a limited number of tissue types. So for example, you have some stem cells, a lot of stem cells actually, in your bone marrow, in the middle part of your bones throughout your body. And these are stem cells. These are adult stem cells. And those stem cells can grow and develop into several different types of bone cells. Yes, there's actually more than one type of bone cell, and also several different types of blood cells. So we're talking about stem cells that can give rise to 2 or 3 different types of bone cells, a couple different types of blood cells, but that's it. They cannot give rise to skin or muscle or liver. Whereas embryonic stem cells can give rise to any tissue type in the body. Now, I mentioned there is a third type, and the third type is called, let me see if I can get the name right, induced pluripotent somatic stem cells. And this is something that doesn't happen naturally, but in the laboratory, scientists are figuring out, and there's a lot of research going into this right now, ways to take maybe bone marrow cells or cells from an adult and manipulate those cells in the laboratory. There are things that you can do to the cells to switch back on the genes that were switched off early in embryonic development. So in other words, you can take cells from an adult human or an adult animal and basically trick those cells into becoming pluripotent again. And you can the ramifications of this, if it can be perfected, and if scientists can figure this out, is you could theoretically take some cells from your own body, have them induced to become pluripotent again, and then those cells could form a liver for you if you need a liver transplant, or a kidney if you need a kidney transplant. So instead of having to rely on an organ from a donor that you are likely to reject, you could actually, theoretically, grow your own liver, your own kidney, your own lung, from inducing your own cells to become pluripotent again. So they're called induced pluripotent stem cells. Would be a third category. But generally speaking, two main categories are embryonic stem cells and adult stem cells. Make sure you know the big difference between them. Adult stem cells are multipotent. Embryonic stem cells are totipotent or pluripotent. All right, and then just a quick point I want to make here, that when, let's say we're talking about embryonic stem cells, just as an example, when stem cells do specialize, and certain genes are switched on and certain genes are switched off, so that those cells will now develop into muscle cells or blood cells or nerve cells or whatever. We're not changing the genes. The DNA code stays the same. In other words, if you were to take a DNA sample from your own muscles, your own blood cells, your own nerve cells, your own heart cells, liver cells, and intestinal cells, and I think you know this, the DNA would be exactly identical in all those cells. So why do these cells look so different, and why do they function so differently if they have the same genes? It's because of which genes are switched on and which genes are switched off. So you take these stem cells and you switch on certain genes and you switch other ones off, and you're going to get muscle. But you take those same stem cells and you switch on a different set of genes and you switch off other genes, and now you grow intestinal cells. So this is what we mean by cell differentiation or cell specialization. It very much has to do with the regulation of gene expression. Okay, this probably goes beyond the scope of what you have to know for AP biology, but it's such an interesting thing, and it definitely relates to embryology. When it comes to triploblastic animals, now remember that's any animal that's more complex than a jellyfish, which is pretty much all animals that you can think of. Animals that have three distinct germ layers: endoderm, ectoderm, mesoderm. All of those animals, from earthworms to insects to amphibians, fish, reptiles, birds, mammals, clams and oysters, starfish, all of those animals can be divided up into two major categories: protostomes and deuterostomes. Protostomes and deuterostomes. And this categorization, like splitting animals into these two major groups, is based on how they develop as embryos, specifically how the digestive tract develops. So I'm going to go back to the blastopore, which was that initial opening at one end of the archenteron, remember? How that develops is different for protostomes and deuterostomes, and also how the mesoderm and the coelom, which is kind of the body cavity, it's what becomes your abdominal cavity and your thoracic cavity in your chest. It's sort of what becomes the hollow spaces in the body, how all of that develops. So I know this is confusing, but look at who we're talking about here. Protostomes would be tapeworms, earthworms, mollusks, which are clams and snails and squids and octopus, and also arthropods, which would be insects and spiders and things like that. That's the protostomes. The deuterostomes is us, chordates, animals that actually have a spinal cord. So that's mostly we're talking about vertebrates there. So fish, amphibians, reptiles, birds, and mammals. But interestingly, it also includes echinoderms like starfish and sea urchins. So you wouldn't think it, but we know from embryology that we are much more closely related from an evolutionary perspective to starfish and sea urchins than to any of these animals. Of course, you probably are sitting there thinking, well, I didn't think I was very closely related to a snail or a bug. But you probably also didn't think you were very closely related to a starfish. And we are in the same broad category of animal, deuterostome, right alongside with starfish. So what does this mean? Okay, so there is a difference in how the mesoderm forms in protostomes and deuterostomes, but the easiest difference to understand is the fate of the blastopore. So if we're talking about a protostome, if we're talking about an insect or a worm, when the archenteron folds in to make this, what will become the digestive tract, the first opening, the blastopore, becomes the mouth. And then eventually, when this becomes a tube that goes all the way through the body, the anus forms later. So protostomes have a mouth before they have an anus. You and I are deuterostomes. So you had an anus before you had a mouth. I bet you didn't know that about yourself. Deuterostomes have an archenteron, they have a blastopore, but that blastopore, that original opening of the digestive tract, becomes the anus, and the mouth forms later. So again, let me go back. Protostomes, the blastopore becomes the mouth. In deuterostomes, the blastopore becomes the anus. So that's the easiest, most obvious difference between protostomes and deuterostomes. You're probably sitting there thinking, well, who cares? I mean, why does this even matter? Well, it matters to embryologists and it matters to taxonomists because, again, this is one of the basic ways we're splitting the entire animal kingdom, is dividing them up based on how they develop, how the embryos develop. Now, there's another difference. Protostomes, when they form remember you have ectoderm, endoderm, and then eventually you have a third layer called the mesoderm. When the mesoderm forms in a protostome, it forms completely distinct and separate from the other two layers. This is called schizocoelous development, and I'm probably doing a lousy job of explaining this because I never have studied a whole lot of embryology. But basically, the mesoderm, which becomes the lining around your body cavities, that forms completely independent of the other two cell layers. Whereas, look at the difference. In a deuterostome, the mesoderm actually begins as an outpocketing of the endoderm. So here's the archenteron, and at some point, little pockets sort of fold out from the sides of the endoderm, and eventually that pinches and separates and becomes a distinct layer, the mesoderm. So if you go back, protostomes, the mesoderm is never really part of the archenteron, whereas in a deuterostome, the mesoderm forms as an outpocketing of the gut or the archenteron. So this is called enterocoelous development. If you want to sound fancy, you can tell your friends that protostomes develop in a schizocoelous fashion, whereas deuterostomes develop in an enterocoelous enterocoelus is probably how you say that, fashion. But the big difference is, again, how the digestive system develops. So protostomes, blastopore becomes the mouth. In deuterostomes, the blastopore becomes the anus, and the mouth forms later. Okay, very quickly I wanted to mention, I don't think you could have any lesson on embryology without mentioning what are called Hox genes. Sometimes they're called home box genes. And this is kind of an interesting topic. Hox genes are kind of like the master switches. They're just a small number of genes that function as like master control switches in the development of an embryo. So there's one Hox gene that kind of starts the development of the head, and another Hox gene might control the development of the eyes, and another Hox gene might control the development of the throat or the chest, another one the abdomen, another one the legs. And Hox genes switch on at certain points in embryological development to basically control the basic body plan so that your legs form where they're supposed to form. You don't want legs growing off the side of your head. You don't want, if you're an insect, you don't want antennae growing from your abdomen. You don't want an eyeball in your back. And so Hox genes control where certain development processes take place. What's interesting about Hox genes is that we're finding, as more and more animal genomes are sequenced, we're finding that different animals have the exact same Hox genes. So this is not the best diagram in the world, but it's the best one I could find in short notice here. The same gene that I guess it would probably be this gene here, maybe. The same gene that activates the development of a fruit fly's head, that exact same gene sparks the development of a mouse's head. Even though a mouse head and a mouse eye look totally different from a fruit fly head and a fruit fly eye, the gene that actually kickstarts the development of the head is the same in fruit flies, in mice, in birds, in humans. So these genes have been conserved by evolution, meaning evolution hasn't really changed these genes much because they're so important for embryos to develop correctly. And that's about all I can tell you about Hox genes. I don't know a lot about them, but I thought it was worth mentioning. All right, the very last thing I want to talk about is a really big step in the evolution of vertebrates. Vertebrates is a group of animals, animals that have a spine, that have internal skeleton, including a spine. And so fish were the first vertebrates. We know that from the fossil record. We know that amphibians were the first vertebrates about 360 million years ago to move onto land. So amphibians were the first land vertebrates, and then of course from amphibians evolved reptiles. From the reptiles you got dinosaurs and birds and eventually mammals. Well, a really big step in vertebrate evolution was the amniotic egg. Fish and amphibians have to lay their eggs in water. Even a frog that is a terrestrial vertebrate has to go into the water to lay its eggs because it doesn't have a way to protect the egg and keep it from drying out. But as you know, reptiles lay their eggs on dry land. And how do they do that? Because they have a very protected egg that has 4 distinct layers. And what's interesting about this is even when mammals eventually evolved from reptile ancestors, most mammals, of course, don't lay eggs. There are some mammals that do, by the way. The duck-billed platypus and the spiny echidna are egg-laying mammals. If you don't believe me, look it up. But most mammals don't lay eggs, and yet mammal embryological development has still retained the 4 layers that are found in a reptile egg. So let me quickly kind of run through. In black, you'll see a description of the 4 layers that are found within a reptile egg or a bird egg. And they are amnion, yolk sac, allantois, and chorion. They each have a function. The amnion is the fluid-filled sac that protects the little baby reptile or the chick. The yolk sac stores the food supply inside the egg. That's where the yolk is found, of course. Excuse me. The allantois stores waste that are built up by the embryo. And the chorion regulates the diffusion of oxygen from the air to the embryo and carbon dioxide diffusing out of the egg as it builds up as a waste product. All right, so notice in red, I've gone through for each of those 4 layers. I'm describing how evolution has tweaked those 4 layers so that they serve related functions, but not exactly the same function in mammals. So now the amnion is virtually unchanged. When you were developing in your mother's womb, you were wrapped and protected inside a fluid-filled sac called the amnion. And that is the sac. The amniotic sac is what ruptures early on in childbirth. You've heard of a mother talking about her water breaking. That's the rupturing of the amniotic sac right before childbirth. So it still serves the same function in the uterus as it does in the reptile or bird egg. Of course, there's no need for yolk for most mammals anymore because the food is supplied, the nutrients for the baby are supplied through the placenta, through the umbilical cord. That's why you have a belly button. So the yolk sac functions as a source of stem cells instead of its food storage function in reptiles and birds. And the allantois, which would have stored waste in bird eggs and reptile eggs, has evolved to become part of the umbilical cord, which attaches the baby's blood supply to the placenta and then connects with the mother's blood supply. And then finally, the chorion, which regulates the diffusion of gases in a reptile or bird egg, has evolved to become part of the placenta. So it's still kind of as an interface between the mother's blood supply and the baby's blood supply so that oxygen can get to the embryo and carbon dioxide can be removed from the embryo. So it's just kind of cool to see that even though we've evolved from our reptile ancestors, we've retained those 4 distinct layers, and evolution has simply tweaked their functionality a little bit. There's a wonderful picture of the amnion, the amniotic sac, which is the fluid-filled sac that protects the embryo. This would be the placenta here, which connects the mother's blood supply with the baby's blood supply. This is just I'll just plug this wonderful video. Y'all know I like Hank Green and his Crash Course Biology. His lesson on embryology is called We're Just Tubes. And he's talking about, of course, the gastrulus stage when your entire body was a ball of cells with a tube running through it. Well, that's 48 minutes. That's a lot longer than I meant for this to be. I appreciate you hanging with me. I hope you're doing well, and I hope to see you soon. Have a great day.